Hello, welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at the core practical that is investigating photosynthesis using isolated chloroplasts. So dehydrogenase is an enzyme found in plant chloroplasts and is crucial in the light-dependent stage. Dehydrogenase catalyzes the reaction of electrons being accepted by NADP, and in this practical, we will be using a redox indicated dye called DC-PIP, which turns colorless when reduced. Hence, it indicates the acceptance of electrons, which is what we are investigating in this practical. So what do we need for this practical? You need the leaf sample, scissors, mortar and pestle, nylon mesh, a filter funnel, centrifuge tubes, ice water salt bath, glass rod, measuring cylinder, beaker, pipettes, bench lamp, buffer, isolation medium, and DC pip solution. So the actual method for the practical is to remove the stalks from the leaf samples and grind the leaf samples in a pestle and mortar and place into a chilled isolation solution. Filter the sample into a beaker using a muslin cloth dipped in isolation medium as filter paper. Place the beaker in an ice water bath to keep it cool and centrifuge the solution at high speed for 10 minutes to separate the chloroplasts into the pellet. Decant the supernatant and add the pellet to fresh isolation medium. So the supernatant is the actual liquid that floats about above the chloroplast. So the pellet is the chloroplast that's left behind. And store the isolation solution in ice. Set the colorimeter to a red filter and zero the cuvette. Again, this is similar to how you would zero a weighing balance, like when you want to measure the weight of something. Same way you're zeroing the cuvette. And zero the cuvette containing the chloroplast, extract, and distill water. Now place the test tube in a rack at a starting distance of 30 cm away from the light source and add DC pip. And take a sample and add to the cuvette. Measure the absorbance of the sample using the colorimeter and take a sample and measure its absorbance every 2 minutes for 10 minutes. And then repeat for different distances from the lamp for up to 100 centimeter again. So in this situation, the distance, not the distance, but the light intensity is the independent variable. That is what we are trying to see the effect of light intensity on how the rate of photosynthesis is affected. So what is the risk assessment? So you are using DC pip and that can irritate the skin and eyes, so it is safe to wear goggles. In case of contact with the skin or eyes, wash immediately with cold water. And then you have biohazard, which can cause allergies from soil bacteria. So it is important to wash hands before you start the practical and after you do finish the practical. And in case of an emergency, do seek assistance. So there can be temporary damage to the eyes if you're staring at the lamps directly. So don't do that. And once again, in case of an emergency, seek assistance. And also you will be using electrical appliances like the centrifuge. So liquids near electrical appliances can act as a conductor. So ensure all equipments are used with dry hands. Once again, in case of accidents, seek further assistance. So what are you going to do with the results? You're going to plot a graph where you have the time on the x-axis, and then you're going to have the absorbance on the y-axis, and you're going to be doing this for every single distance you've measured from. So for example, your first graph is going to be time sorry, the absorbance being compared against time for 20 centimeter, and then you're going to do the same 40 centimeter, 60 centimeter, and so on until 100. So, what conclusion should you be expecting? So, you should see that as light intensity increases, the rate of photosynthesis also increases. And this is because higher the light intensity, the higher the rate of photoionization of the chlorophyll pigment. Hence, the overall rate of the light dependent reaction is faster. This means that more electrons are released by the chlorophyll, hence the DC pip accepts more electrons. Hence, with decrease in distance, the less time it takes to go from blue to colorless. When the DC pip is blue, the absorbance is higher. The activity of the dehydrogenase enzyme can be determined by the rate at which the absorbance decreases. So higher rate of decrease, which you can see from the steep gradient on your graphs that you plotted before. So a steep gradient would indicate that the dehydrogenase is highly active. And then a more shallow gradient would suggest that it's not as active. So that is all from Professor Snap today. I hope you found this video useful, but I would recommend going back to watching the more detailed video on the actual process of photosynthesis. So watching the theory side of it and then going back to the practical would make a lot more sense. So I would really recommend you do that. And that is all for this video. And don't forget to like, share, and subscribe.